Hello and welcome to the CVL podcast, coming to you from the UT Dallas Center for Vital Longevity, which is dedicated to pursuing research that leads to cognitive health for life. Our goal is to educate you on the latest matters relating to cognition in the brain, and with each episode we'll be interviewing leading scientists and sharing their insights and suggestions to promote cognitive health throughout the lifespan. My name is Amber Kidwai.
And my name is Sarah Munir.
And this is the CVL podcast. In this episode, we meet with Dr. Gagan Wig, an associate professor at our very own Center for Vital Longevity, where he's also the director of the Wig Neuroimaging Lab, which is a cognitive neuroscience lab focused on understanding how and why the brain ages. He obtained his PhD in cognitive neuroscience from Dartmouth College, and then he was a postdoctoral fellow at Harvard University and then Washington University. Dr. Wig's lab has produced some groundbreaking research in understanding the organization of large-scale human brain networks and how these change over the adult lifespan, with a particular focus on memory and attentional processes. We begin this episode with a description of the connections between brain regions, how they communicate and coordinate in what we call brain networks. Brain connections can be anatomical or physical connections where fibers or bundles of white matter are physically linking different regions of the brain. But brain connections can also be functional. This is when different regions show coordinated activity. Think of complex brain functions like language, emotion, or attention as a symphony, not a solo performance. There's no single language area or emotion center in the brain. Instead, these functions rely on multiple brain regions working together. And thanks to cutting-edge imaging technology, massive amounts of data, and incredibly powerful computers, we can now create a digital 3D map of the human brain. These brain maps are revolutionizing our understanding of how information flows through the brain, allowing researchers like Dr. Wig to gain new insight into how our minds work and opening up exciting possibilities for improving brain health. In today's episode, Dr. Wig shares some fascinating insights in this conversation, including the fact that brain networks are segregated into clusters or nodes that coordinate their activity with other nodes. And we learn how brain networks that are more segregated are linked to better cognitive function, including memory performance. I was fascinated to learn that as we age, our networks can actually become less segregated. Brain networks develop throughout our lifespan, and we discuss the impact of a person's environment on their brain networks, as well as the effects of aging and the effects of neurodegenerative diseases such as Alzheimer's. So join us as we delve into the world of nodes and edges and discuss the fascinating connections throughout our brains.
Dr. Wig, thank you so much for joining us today. We always like to learn a bit about our guests before we get into the science. So could you tell us a bit about your background and what led you to this area of research?
Yeah, sure. Thanks for having me. I'm a cognitive neuroscientist. I did my undergraduate degree in biopsychology and in business. And throughout that, I started appreciating how interesting it was to study the brain and to study psychology. And I switched from being a pre-med to someone who really wanted to just do research. So I went on to pursue my PhD in cognitive neuroscience. I focused on human memory systems. And from there I went on to move to pursue a postdoctoral fellowship where I studied human aging and memory. And then since that time, I've been thinking a lot about large-scale brain networks and have been applying that training to my work here at UT Dallas.
Okay, so why did you choose to focus your research on brain networks, and what do you think makes this approach particularly valuable for understanding the brain?
Yeah, I mean, I think we've learned a lot about the aging brain, both in terms of its function and its structure, and how those properties support cognitive processing. For me, the appeal of studying brain networks is that we know that the brain operates as a network, much like other networks in society and in technology. So given that the brain and cognition are supported by the interactions across regions, it makes sense to me to study how those things break down in the same context, right? In the context of how networks change over time and how the networks change as we age.
Yeah, super cool. So staying on the topic of brain networks, could you break down the concept of brain networks for our listeners? So specifically, what are the basic units or components that make up a brain network? And how do researchers like yourself identify and define these networks in the brain?
Yeah, so the study of a network involves describing the network first with a mathematical framework, and it's pretty simple conceptually. A network requires you to have things and then interactions amongst those things, and those things can be technological, like computers. They could be social, like individuals, and they can even be biological, like proteins. And then the interactions could be wires, or they could be relationships, or they could be functional relatedness. So in the brain, there are multiple levels at which we could describe a brain network. The obvious one that I think most people think about are neurons and how neurons are interconnected. Our neurons are a basic computational unit of the brain, and they're all highly connected with one another. So at that level, the network would be neurons as nodes, and then edges would be the axons and dendrites, so the connections shared between neurons. Moving up in scale, though, you can describe brain networks at the level of circuits or columns, which are sets of neurons that are doing similar things, or larger groups of neurons or larger groups of circuits or columns that form areas. So I think most of us, when we think about brain networks and we're looking at brain network data in cognitive neuroscience, we're focused on this latter scale of analysis because our imaging technologies allow us to image that scale of resolution, that spatial scale of resolution. So for us, the nodes are brain areas, which are roughly a few millimeters to a centimeter large, and then the interactions would be how those areas interact with one another, and those would be the edges. So that's how we describe a network, and then ultimately we're trying to understand how that set of interactions bring out more complex patterns of organization and kind of give rise to cognition.
So when we talk about the brain, we often talk about structure or anatomy versus function. When it comes to brain networks, what's the difference between an anatomical and functional brain network? How does the functional network differ from the physical or anatomical connections in our brain, and why is it important to understand both types?
Yeah, it's a good question. I think when people hear about brain networks, the first thing they think about are anatomical brain networks, right? Which would be, again, focusing on the areal level. They'd be composed of those areas of the brain, and then the wiring between those areas, right? So large fiber tracts of myelinated axons. But they're essentially these large fiber cables, these physical structures, and that's an anatomical brain network. It's a physical entity. A functional brain network, it has some components that are similar, so the nodes are similar, right? We're talking about areas. But in the case of a functional brain network, the interest is in describing how those different nodes or areas are functionally related or how they're interacting. And the interesting thing is that two regions need not be directly anatomically connected for them to be interacting, right? They could be interacting via a third intermediary region, or they might be receiving a common input from another region. And I think it's an important and separate and informative way of thinking about interactions. So, in the same way that a brain network might be described via its structure or its function, there are other types of networks as well where you might have a similar distinction. So, the internet is composed of computers that are connected via physical wire cables. But the internet gives rise to a set of interacting web pages, and you can describe those web pages, which might not be physically connected, but they might be linking to one another via their shared information. So there's just two different ways of describing, a common system. And those two different ways give rise to interesting features that would each be equally informative and worth investigating in their own right.
Got it. Super interesting. So kind of staying with the topic of going to the functional networks. So we know that there are these functional networks that can be active when individuals are engaged in certain tasks, but we can also measure what's called resting state networks, which, as the name implies, these refers to the connections or co-activations of certain brain regions when a person is at rest. And a lot of your work actually focuses on resting state networks in particular. Why did you choose to focus on resting state networks? Could you kind of describe what made you interested in that specifically?
Yeah, so I started my training analyzing patterns of brain activity in response to physical, presented stimuli or given task instruction. So as you described it to be a task-related set of patterns. But what was interesting to see is this emergence of a separate way of describing brain function. It turns out that even in the absence of a task, the brain is very active. I think we all know that. But the interesting observation, which was made by Bharat Biswal in the 90s, was that if you look at the brain in the absence of a task, right, so someone's laying in the scanner, just doing nothing, the activity is highly informative and it contains structure, or I guess organization. So parts of the brain that are functionally similar tend to be active in similar ways. Their patterns of activity are similar. They're correlated. So that could be appreciated by looking at the blood oxygen level dependent signal, the BOLD signal, which is what we measure with fMRI. So parts of your brain that are involved in motor function and motor planning are all highly correlated. Again, even when you're not doing a motor task per se. Likewise, parts of your brain that are involved in executive control and executive function, those are highly correlated. So what that revealed to us is that there's this organization, this intrinsic organization of brain function that's detectable, even in the absence of doing a task. And I think that has a lot of appeal because it allows you to interrogate the functional organization of the brain and how it might change without having people even do a task per se. And then there's various reasons why you might want to do that.
Mm-hmm. Yeah, I was kind of reading some of your papers, and you kind of phrased like the resting state functional connectivity. You said that it's hypothesized to reflect these statistical histories of coactivation patterns. Could you kind of elaborate on that?
Yeah, so these coactivation patterns, they don't come out of nowhere, but the remarkable thing is that you can see them evolve over development and, of course, in aging, as we're seeing now. But what they're reflecting, what we think they're reflecting, is the statistical history. Right? So we all have experience with the world, and we're interacting with the world and objects and people in different ways. And these coactivations or these correlations, they are the product of those histories. So to the extent that different parts of the brain are doing things at the same time over your life course, they'll be highly correlated at rest, which is why you get these motor circuits and language circuits and visual circuits and so forth.
Yep. So could you explain the concept of brain network segregation? So what does it mean for brain networks to be segregated, and how does this relate to the idea of different parts of the brain working together?
So, when you look at the resting state function, right, or these correlations, or these functional networks. As I said, they're not random. Parts of the brain that are involved in motor function are all highly correlated. Parts that are involved in vision are highly correlated, and so forth. And now, rather than just looking at a given region or a small set of regions, you could look at all the regions in aggregate and see whether and how they might form, aggregated clusters, right? Much the same way that you would do with a social network. For any of us, we have different groups of friends and different social communities that we interact with. There's an organization once you look at all the sets of interactions of your relationships. And when looking at the brain in this way, it turns out that across the brain areas are these groupings, right, where sets of areas that are involved in given functions are highly correlated. And we refer to that as a modular organization. And that modular organization is a property that's evident in many organized networks, again, across technology and biology and social systems. And it's important because what it confers to the network is a form of functional specialization, right? So having tightly interacting nodes, or in the brain's case, areas that are less tightly interacting with other groups of nodes allows those nodes to be functionally specialized, and it permits them to do specialized processing roles. So we describe that organization, that modular organization, mathematically via a measure called segregation. And in a healthy young adult brain, the brain is segregated. So you have groups of these areas that are all doing different sets of things, and they're segregated from one another. That doesn't mean that they're disconnected from one another or that they don't interact, but rather that they're allowing for functional specialization while simultaneously supporting cross network interactions.
Got it. So again, just to kind of clarify, so these kind of little groupings, these what you would consider like subnetworks within this like larger scale network, right? So these subnetworks are each specialized in their own little thing.
That's right.
So we all know that our brains change as we age. And you mentioned the functional specialization and how young brains have segregated systems, and that makes them more resilient to particular types of disruption and to damage. But how do our networks change, our brain networks change as we age, and how does that relate to changes in our cognitive abilities as we age?
Yeah, so what we're finding, and we reported this around 10 years ago here at CVL, was that when we look at individuals of different ages, adults in particular, ranging from 20 to 90, that pattern of segregation is different across individuals and in a very systematic way with respect to age. So as I said, with a young adult brain, you see highly segregated networks, which as we've discussed, is supporting functional specialization. But with increasing age, that segregation decreases. So those subnetworks or those modules or those brain systems become more and more blurry. So it's a form of de-differentiation wherein that specialization is lost as revealed by the patterns of functional interactions. So regions of your brain that are typically not interacting with other regions that are less functionally related tend to interact more with those other regions, or at least they show more similar patterns of activity over time.
So does it that desegregation or despecialization that you mentioned, does that then mean that those people who have less segregated networks have worse cognitive abilities, or is it the other way around? Is it better to have less segregated networks as you age?
It doesn't mean that per se, and we were uncertain of that until we started looking more closely at cognitive function. And it turns out that individuals that have less segregated networks do have worse long-term memory function. But that's not just purely an aging phenomenon. That's also the case with younger adults, right? So there's younger adults that have lower system segregation, this measure of specialization of the network, and they have worse memory function than you would expect by average. Likewise, there's older adults who have higher system segregation, and those older individuals tend to have better memory function. So we think it really is supporting cognitive operations and supporting at least long-term memory performance in an interesting way.
And again, like when we're talking about this decrease in segregation with older age, we're still referring to those resting state connectivities, right? So I'm guessing if you would measure these connectivities when somebody's doing like a task performance, that's a different picture, correct, with age?
That's right. That's a good question. So this is what we're describing here is the intrinsic organization, the organization in the absence of overt task demands, and we think that provides this like scaffold or this backbone on which cognitive operations rest. So when you're actually doing a task, you're reconfiguring that resting state network. When you do a task that involves linking visual information to attention systems, regions of the brain that are involved in visual processing and attentional processing, they're more tightly intertwined. Mm-hmm. So that's a reconfiguration, right? So what this resting state network is essentially your baseline from which that reconfiguration emerges. The reconfiguration we think is tightly bound to what's happening at rest, and it's limited and it's also promoted by resting state organization.
Got it. So you've done some really groundbreaking research looking at the factors that may influence brain network organization. Could you describe some of these factors? So specifically, I'm referring to like socioeconomic status that you've explored. So can you kind of delve into that?
Yeah, I guess I can start by saying, no two individuals exhibit the same pattern of brain aging. So if we look at these brain networks that at rest over time in an individual, meaning a longitudinal observation, two people could start at the same point, but they might traverse very different trajectories. So the big question for us has been, what are the factors that might lead to accelerated brain network aging in one individual, yet result in maybe maintained brain network changes or the absence of brain network changes in another individual? We know there are a lot of health, lifestyle, and environmental factors that result in accelerated brain aging and differential health outcomes. There are disparities in brain health that are tightly linked to exposures and to our environments. So that's been the first place we've been looking at to understand whether or not we can start to pull apart why one individual might exhibit a pattern of brain aging that looks to be more accelerated than another individual. So for this, we have ideal ways that we would want to approach this work, right? We'd want to understand, your health, your nutrition, your sleep habits, your exercise, your mental stimulation. There's lots of things we would want to learn. But as a first pass, what we've been doing is using a measure that is highly correlated with those important health and lifestyle factors. And that measure is your socioeconomic status. So your socioeconomic status, which often refers to your income or your educational attainment or your occupation as an adult, it's highly correlated with many of these health and lifestyle measures. So we've been using socioeconomic status to understand whether or not SES or socioeconomic status is stratifying individuals and whether it could predict who's going to decline and who will not. Mm-hmm. Now, SES isn't it's not deterministic of your health and lifestyle, right? There's lots of variation, but it seems to be a reasonable course proxy for exposures. People that are lower SES tend to have higher risk of not only brain disease, but also heart disease, right? Because they're being exposed to adverse environments on average to a greater extent. They have more limited access to necessary sources of health care and even food, right? They would live in there's many food deserts that exist in lower SES environments. So for us, that's been the first way of tackling this critical problem of understanding disparities in brain health.
Got it. So kind of based on your research, have you noticed that individuals coming from lower SES backgrounds have more desegregation, for example? Have you done studies on that?
That's exactly right. So, we've again looked at this in a few different ways. The first way we looked at this was by just comparing individuals from lower SES versus higher SES. And it turns out that they do stratify with respect to these measures of network organization. Lower SES individuals exhibit lower brain system segregation. When we look at it longitudinally, more recently, we found that individuals that are absent a college education, which is again an indicator of lower SES, seem to show more accelerated patterns of brain network decline. So these are all signals to us that we're at least examining a measure of brain function that is paralleling some of the epidemiological observations related to SES and health outcomes.
So with that in mind, do you feel like there's anything we can do to improve or maintain our level of brain segregation as we age?
It's a great question. So I guess I'll first start by saying, I mentioned this observation with the college degree attainment. It's certainly not the case that having a college degree is conferring you a guaranteed advantage. What we're suggesting is that the college degree is affording access to something for some individuals that is absent in the individuals that don't have a college degree, right? So there's a correlation between college degree and income levels and living in better neighborhoods, and having access to critical resources that are that we all deserve, but not everyone exhibits the same pattern, right? It's just there's a group effect where individuals in one group are more likely to exhibit a pattern of desegregation than individuals in another group. So following that, now we're trying to understand, well, what is it, right? We know it's not having this diploma in your hand, but there has to be something else. So we're trying to understand the specific health and lifestyle factors that might lead to more accelerated patterns of brain network decline. At this point, I think it's too early to say, what things might reverse it or halt it or slow it down, or whether that's possible. But we're really trying to interrogate, what are the specific components that might matter most for brain aging.
So we touched on age-related changes in brain network segregation, and I'd also like to touch on disordered aging, specifically Alzheimer's disease. So how do brain network changes in Alzheimer's differ from normal aging?
Yeah, so we've started now examining Alzheimer's disease, dementia, and using the same framework of understanding brain network changes. We know Alzheimer's disease to be this progressive neurodegenerative disorder that is impacting specific circuits of the brain. But we also know that as the disease progresses, it does, individuals who are suffering from dementia have broad changes in cognitive function and behavior. So examining their network interactions is a very strong way of interrogating, what it is that might be changing in the brain. So when we apply the same framework to patients with Alzheimer's disease, what we've started to do is look at, again, individuals who are varying in dementia severity and trying to understand whether or not patterns of brain network decline in those individuals differ from healthy aging. So this work has been led by both Zhiwei Zhang, a doctoral student in the lab, and Michaela Chan, who's a staff scientist in the lab, who collectively have been looking at various data sets that we've had the privilege of getting access to understand patterns of brain network decline in Alzheimer's disease. And what we've been finding is that, first of all, if you look at healthy individuals, longitudinally, individuals who decline a lot in terms of their segregation are at greater risk of prospectively developing dementia. So that's a finding that Michaela Chan published a few years ago. And then second, if you look at individuals with varying degrees of Alzheimer's disease dementia severity, there's also an impact on this measure of segregation that's independent of aging. So individuals who have greater dementia severity show lower segregation independent of their age and what their segregation is as a function of their age. So there seem to be two separate sets of processes operating on your network, one of which accompanies just healthy aging, and the other of which is probably a consequence of neurodegeneration and pathology that's associated with Alzheimer's.
So are there any promising interventions being studied to maintain healthy brain networks as we age?
I think a lot of people are exploring various ways to slow down, the brain aging, and analyzing brain networks is one way of understanding whether or not your intervention has been effective. I think the types of interventions that have been studied so far include exercise interventions, cognitive training interventions, and then skill learning. But I think the jury's still out yet as to whether or not any of those are going to arrest or slow down patterns of brain aging and even cognitive function more generally.
So do you ever actually see, for example, there's an intervention and segregation can be encouraged? I mean, can it ever go back, or is it like once you've desegregated those networks, it's going to stay like that? Or is it that if you changed your lifestyle and had a more healthy lifestyle, potentially you would just be maintaining what you already have, or would you be able to improve?
Yeah, that's a great question, right? Whether or not a health and lifestyle intervention might reverse or slow down, I should say, patterns of typical brain aging. We haven't seen that yet. We haven't tried to do that specific study in our lab yet. I know there are several laboratories that are starting to explore that area. What I can speak to is observations based in patient-based research. So patients that have suffered acute strokes or lesions or have had tumors resected, their brain networks change pretty drastically in the initial period, along with cognitive function in some cases. But then the recovery of those functions is often associated with a return in measures of segregation. And
I was just thinking about you were talking about segregation of networks and how it's kind of based on our exposure as we grow up. Our exposure to the environment causes certain networks to be created in a certain way, and those are segregated. So it just made me think about kind of disordered networks, like people who have pathology, some kind of psychopathology, other than dementia and Alzheimer's, but they may have gone through something in their upbringing which caused network, I don't know if you'd call it disorganization, or not segregated in the way that would be in a healthy person. I know this isn't exactly your field, but do people look at the relationship of functional specialization of these networks and how that relates to psychopathology, and if there is changes in those networks when people are recovering from symptoms or improving?
Yeah. So it's certainly an approach that's being examined in the context of psychopathology and psychiatric disorders. And there's some evidence that there are ways of better understanding psychiatric disorders and psychopathology via this network-based lens, because again, those are disorders of large-scale network interactions. But I think I also want to focus on something else you mentioned in terms of how things start out, because that's another part of what we're doing. And I think the nice thing is that it blends some of the work that's now coming up and having and being established at CVL, which is examining the early life stages. So, while it's true that our exposures are altering our organization of our networks, and we've seen that in adulthood, we also think that those networks are established, of course, in early years. And the big question for us is whether or not we can disentangle different components of the life course and how they might contribute towards not only the level of your network, but also how it might change. So the current effort in the lab is to get a more fuller understanding of the life course, to understand how early childhood exposures and adversity may lead to set points that from which you start to change over time. And I think those are going to be critical towards understanding patterns of brain aging and how they might differ.
Yeah, I know definitely in terms of general cognitive abilities and memory changes over time, research has kind of shown that, the changes that are shown in late life that we kind of study when we look at cognitive aging, sometimes those are evident at a younger age. And I think, like you said, that push towards looking at more of the lifespan is quite crucial. And even looking at kind of middle age, where things start to change before people kind of reach older age, and looking for those earlier signs of change or desegregation in the case of networks might also be quite useful.
That's right. Yeah, a lot of the things we're doing as well are focused on understanding patterns of brain aging prior to what's typically considered older age adulthood. Yeah. Mm-hmm. So exactly as you say, focusing on midlife, preclinical stages of an individual's life course, just to understand if there's risk factors that might be identified in terms of their impact on the brain. I think that's where a lot of us need to move towards in order if we have any hopes of slowing down the aging process.
Because a lot of those changes typically begin in middle age, but you just don't see them behaviorally, meaning in somebody's behavior, until they reach old age, which may be 20 years later. So I think it's really interesting to look at what's actually going on, and like you said, in those preclinical stages. Yeah.
So kind of going off of that, how in early life, coming from a high socioeconomic status can really predict how your health is going to be, your cognitive health in older age. So do you think that there are public health policies or interventions that can be implemented for like this younger age group before we're seeing these clinical symptoms that we can implement that could protect against these brain network desegregations and so on?
So I don't think we know what the data are going to look like yet in terms of network declines, right? We're in early stages of characterizing life course brain network changes. But I think the evidence is clear that there are things that can be done at the societal level that would promote lifelong health, and they start in early years, which they include access to adequate and strong education, but also sufficient sources of nutrition, safety from violence and adverse events, and growing up in enriching and enriching environments that foster healthy living.
Yeah.
Okay. So thank you so much, Dr. Wig, for taking the time to have a conversation with us. Where can our listeners find out more about your research?
Yeah, well, thanks so much for inviting me to chat. If anyone's interested in learning more about our research, we feature most of our research on our lab webpage, which is accessible via the Center for Vital Longevity website. And then, of course, feel free to reach out to me directly if you have any other questions.
Yeah, okay. So we look forward to hearing more about your future work. Thank you for listening to the CVL podcast. You can follow us on Spotify or look for episodes on our website, where you can also learn more about the Center for Vital Longevity. Just look up cvl.utdallas.edu, and here you'll find information about our research, as well as information on how to volunteer to participate in our studies and how to donate to support innovative research to understand, protect, and enhance the vitality of the mind across the lifespan. Join us next time as we continue to explore the latest research and strategies for keeping our brains healthy throughout our lives.